Hello, pleasure to be here. Pretty big event. There is another big event in a couple of months in Europe, the Olympics. So the question I'd like to address now is a bit similar to this one. So imagine you qualify for the Olympics. Would you rather train, prepare for what's to be done there, or go to bed and wake up on the starting line? So I think here we're kind of all trying to transition the world from chemistry towards biology. And if we think about how the world really works, to mention a great book, well, synthetic nitrogen fertilizer are really on top of what's needed for modern civilization, alongside energy. I mean, they feed half the world. By the same token, they generate severe negative environmental externalities. So there's an opportunity with this nitrogen crisis, again with the microbiome, and we can use bacteria to promote plant growth, but also bring other agronomic benefits, including a reduction of their addiction to fertilizers. So I'm sure in this room you're all familiar with the way these bacteria are made. What we do is we change a couple of things. We alleviate the need for large-scale fermentation, and we don't do long-term stabilization. What we add is a step of amplification on-site of the bacteria, what we call fermentation. So if you've been to a hotel room recently, you know these coffee machines where you put a capsule. So that's very similar conceptually to what we use. A simple device with single-use consumable for aseptic transfer. A device can treat about 400 acres. We can replicate it in an array, and this way we can deliver fresh bacteria. So here is the exciting part. I'll talk about three examples. We have a new process, so we unlock novel biology. So the first thing, and I think I appreciated the M&A announcement from this morning, is we can now use bacteria, requalify bacteria that were previously discarded in any screening because they were great, but they had poor manufacturability. They were difficult to stabilize. We can include them in our process and productize them. The other is like, if you think about the bacterial growth curve, we have bacteria that are actively dividing, not in lag phase. So when the next challenge for them is to enter the plants through stomata, they have a limited window of opportunity. So we feel we prepare them the best for what they need to do. Third one is on the media. So there's data showing that growing the same bacteria in two different media, you can get different metabolism in a subsequent media, both for phosphate solubilization in an in vitro assay, but even agronomic effect can be different. In one case, the same bacteria will lead to more heavier roots or heavier shoots. So obviously, we are generating finer grain data with the great teams and the great tech at Ginkgo. But I think already the ability to access this subset of parameters that's not accessed now should enable us to make the best performing bacteria and win. Obviously, this is amenable to data-led personalization. My name is Julien. The company is called OneOne Bio. I'll stop here. I think human metabolism is quite important, and it's time for lunch. Thank you.